Hi everybody, and welcome to the top 10 things to know about SARS-CoV-2. Number 1. COVID-19 is the name of the disease. The disease is caused by the most recently discovered coronavirus, severe acute respiratory syndrome related coronavirus, SARS-CoV-2. A virion is a complete infective viral particle consisting of the RNA of the virus surrounded by its envelope. Fomites are inanimate objects like doorknobs that can be contaminated with virions and then give them to you. So in this case, the classic example is if somebody sneezes on their hand, uses a handrail, and then you come along behind them and use it afterwards. The coronaviruses are a group of related viruses that cause diseases in mammals and birds. There are about 40 of them, and they are not new. They were discovered in the 1930s in domestic chickens, and the first human coronavirus to emerged in the 1960s. SARS-CoV-2 is the 7th coronavirus to emerge. They're very, very closely related. This is very good. You can see on this phylogenetic tree just how closely related they are. Here are all the SARS-CoVs, and the ones with red text, SARS-CoV-2 and SARS-CoV, are the ones which are able to infect humans, and they share about 78% of nucleotide base pairs. So this means that what we know about SARS-CoV is probably true for SARS-CoV-2 as well. The next thing you need to know about them is that they have absolutely enormous RNA. The reason this is good is because it's very easy to detect. So here's the spike glycoprotein, that's the naughty bit that attaches the virus to your cells. Here's the envelope membrane protein, and here is the membrane protein. The three of them are embedded in the lipid bilayer, this grey circle going around the cell, and this is what makes it so fragile. Soap destroys the lipid bilayer, and that enables us to kill the virus. This blue squiggle is the biggest strand of RNA yet found in a coronavirus. It's about 30 kilobase pairs, and the reason it's so good that it's large is that it makes it very easy to detect. People infected with SARS-CoV-2 shed the virions and pieces of viral RNA from broken virions in their bodily excreta. Whether they have symptoms or not, they shed it, and even if they test negative for COVID-19 with a throat swab, they shed it. People that do have symptoms carry on shedding virus RNA and virus particles themselves for 7 weeks after they recover, if they become ill. All of that stuff goes in wastewater. We flush it down the toilet. So wastewater and receiving bodies have a significant role as potential sources of epidemiological data and also as factors in public health risk. Coronaviruses haven't been waterborne in the past, so the standard virus concentration and detection methods we use for viruses in water don't work very well for enveloped viruses. Because of this gigantic RNA, we're seeing a lot of work coming out now detecting the presence of SARS-CoV-2 RNA in wastewater, but that doesn't mean that there are infective virions in that water.